Hi everyone, I'm Vishal. I'm a third year PhD candidate in immunology in Dr. Camilla Coelho and Goran Baich's labs in Mount Sinai. And today I'll be talking to you about my PhD work so far on basically identifying how somatic hypermutations can be indicative of epitope immunodominance in antibody repertoires. So we all know that given a polyclonal set of B cells that have their own germline backgrounds that are targeting an antigen, there are certain regions of an antigen that attract more antibodies in what we call immunodominant epitopes. While there are also certain regions that do not attract as many antibodies, or in what we call atypical epitopes. Now, these can be features of the antigen, be it accessibility or certain surface features that would determine why certain antibodies target these epitopes more or less. But there are also certain features of the B cell itself that bias recognition of particular epitopes. And by features, I mean mostly genetic features determining the antibody variable region. So the obvious question that comes up is if there is any association between such features of the B cell receptor and immunodominance of the cognate epitopes targeted. This has been widely studied across multiple different antigens, which we have detailed in this review here, where there are clear associations of certain B and J genes with specific epitopic regions across different antigens. But there is another thing that adds complication to this process. We all know that given a polyclonal set of BCRs that are germline, these are unmutated as of now. They also enter germinal centers where they undergo affinity maturation. So here they acquire mutations that specifically increase affinity towards their cognate antigen. And as an output, you see these BCRs with now somatic hypermutation that bind with higher affinity to their cognate epitopes. So a question that arises here is, are there certain features of SHM that can inform the cognate epitopes immunodominance? Because the affinity maturation in a way is also proceeding specifically towards that particular epitope. Um, so here we define a certain feature of somatic hypermutation that we hypothesize will help us sort of correlate it with epitope immunodominance. So given VD-J recombination, we all know that there are numerous possibilities of different VD and J gene segments recombining to form a single VD-J exon. But we also know that given this primary diversity, there are some V genes that are often utilized by antibodies across different specificities. One of the most common examples you see in the human repertoire is this V169 V gene that's used by antibodies against flu, HCV, HIV-1, and many other viruses. So let's, for one, consider such a highly functional V gene, X, and let's say we have an antibody clonotype specific to antigen A. So here you're restricting the V, D, and J genes. Um, and on the other hand, let's say you have these three categories of antibodies against antigens B, C, and D that still use the same V gene but can have their own junctional gene pairings that render specificity to those particular epitopes. On one hand, we define clonotype restricted mutations as those that are predominantly enriched within the clonotype specific to antigen A. And on the other hand, we have clonotype shared mutations that pretty much occur in an epitope agnostic fashion where they have similar frequencies in occurrence across these different antigen groups. Um, and going back to our hypothesis, we claim that these clonotype shared mutations are those that mostly help your affinity mature towards immunodominant epitopes, while clonotype restricted mutations are more so required to gain access to these rare atypical epitopes within an antigen. And here today I'll be talking about briefly two stories, and the first one will be focused on the clonotype restricted mutations. So this study came out recently last year in Cell Reports, where we identified a public antibody seen in multiple individuals called M15. And the way we did this was firstly we identified this M15, the original antibody, in a cohort of XBB mRNA or protein-based vaccines. And we mapped the sequence of M15 based on its V and J germline gene identity and CDR3 similarity that we assigned using assigned genes of changeo. To three cohorts and databases. There was another cohort of mRNA vaccines, CoV-AbDAb, which is a database of SARS-CoV-2 specific antibodies, and this dataset called OAS or Observed Antibody Space that has deep sequenced paired heavy chain, light chain repertoires from SARS-CoV-2 infection and all these other categories. We interestingly see that we find M15-like sequences exclusively elicited upon SARS-CoV-2 exposure, and from here we had a total of 147 M15-like sequences from which we could identify our clonotype-restricted mutations. So to do that, we firstly identified convergent mutations that are seen in at least 3 individuals out of the 7 we had, and we compared the frequencies of these convergent mutations between the 147 and 15-like sequences we initially identified and these other three antigen categories. So these were basically non-S2 antibodies from CoV-AbDAb, influenza, and malaria antibodies that use the same V gene as M15, that is V459 for heavy and KV320 for the light chain. When we compared the frequencies of these convergent mutations, we see that in the heavy chain, you don't really see much of a difference. They're all occurring at similar frequencies, but in the light chain, some of these tend to pop up more in the M15-like sequences. So there were at least 5 mutations here that had significantly higher frequencies in M15-like sequences compared to these other three categories. So we, of course, hypothesized that these might be the key players in affinity maturation of M15 itself. So we reverted these mutations back to the germline to see if they had a significant effect on affinity. We chose the three most convergent mutations here, and when we reverted each of them individually, we didn't really see a significant decrease in affinity. But when we took out all three of them together, the affinity got down to the level of the UCA or the germline version of M15, implying that these three mutations do play a key role in maturation of M15. So to further validate this, we obtained a structure of M15 in complex with S2. And here as well, you clearly see that these residues in yellow, which represent these clonotype-restricted mutations, indeed form contacts with S2. What was also interesting was this revealed that M15 binds the cryptic epitope in S2 that hadn't been identified before. Uh, Basically, M15 is able to open up the S2 trimer and access an internal epitope, which has a hydrophobic cleft. And now these three mutations, interestingly, were all polar to hydrophobic mutations that helped M15 access this highly hydrophobic cleft within S2. So this is something we're investigating more in other antibodies to see if we can identify more clonotype-restricted SHM-driven recognition of cryptic epitopes. So in the interest of time, I'll move to the other part of the project, which is on the clonotype shared mutations. So for this, it's mostly ongoing work where we're collaborating with Dr. Ali Alivedi from WashU, um, where we're looking at the spike-specific germinal-centered B cells from SARS-CoV-2 mRNA vaccinees. And from here, for each V gene, we have identified clonotype shared mutations in the y-axis here that are shared by at least 5 clonotypes. Represented in purple here. And what you can clearly see for these two V genes I've showed is that these mutations are indeed really common across these different clonotypes. So given these two V genes are so common in the repertoire, and we have so many SARS-CoV-2 antibodies documented in literature, we asked, given structures of these antibodies, can we look at what roles these mutations play in general when it comes to antigen-antibody interactions? So um, we looked at CoV-AbDAb again, which had these SARS-CoV-2 specific antibody sequences, specifically looking at these 330 and V169 antibodies. And for each of these mutations, we first annotated whether the mutation occurs or not in each antibody, which is a column here, and whether the mutation is a contact with the antigen, which is orange, or the light chain, which is in green. And what was interesting was these mutations are not only common across different antibodies, but they also seem to have similar functional roles. For example, S31N here seems to always be a contact with the antigen, while Y95F, whenever it's present, always seems to be a contact with the light chain. So this was interesting, and we asked, okay, if this is true for different epitopes within Spike, this might be true for other antigens in general that use for antibodies that use these V genes. So we did exactly that. We looked at this other database of again antigen antibody structures um, against multiple other antigens, as you can see, influenza, Zika, HIV, a lot of other viral antigens. And here we again see similar pattern where depending on where the mutation is positioned, it seems to have conserved functional roles, be it contacting the antigen or contacting the light chain. So Till now, we were looking at this from the lens of mutations we identified in the spike repertoire. But what if there are mutations, such pan-antigenic mutations in general for these V genes across different antigen specificities? So we went back to this database SABDAB again, where we looked at these antigen antibody structures, and I'm showing you only the V169 antibodies here. And we see a lot more of these mutations seen in at least 10% of these antibodies that are clearly shared across different antigens. So each column here again is a mutation, and the different colors represent how many different antigens they're shared across. Since we had the data of antigen antibody structures, we could also look at contacts to see if these mutations were also forming contacts with the antigens. And here, what we also did was for each antibody, we looked at whether the residue, if it's mutated, is it a contact? But if it's not mutated, is the germline a contact as well? Basically, to see how often does the mutation create a de novo contact beyond the germline residue. And what was interesting was when we segregated this into heavy chain, light chain, and antigen contacts, we don't really see a different, significant difference in frequencies between germline and pan-antigenic mutations, meaning that more often than not, these pan-antigenic mutations aren't really inducing de novo contacts, meaning the germline residue at that position was already probably a contact, and the mutation is either increasing affinity or sort of maintaining neutral affinity while being a contact at that particular residue. So, in conclusion, so far we've sort of identified this public antibody M15 that uses these clonotype restricted mutations to affinity mature towards um, a rather atypical cryptic epitope, the central interface epitope on S2. And on the other hand, we have identified these clonotype shared mutations that are often panantigenic not only in occurrence but also their function when it comes to antigen antibody interactions. And we also show that they rarely contribute to new contacts in antigen antibody interactions. Now, this is still the tip of the iceberg, and we're digging this in further to see what role these panantigenic mutations can play when we introduce them to different germline backgrounds. Are they sufficient to improve affinity for different germlines against different antigens, or are they mostly neutral bystanders? Uh, on the other side of the story, again, we're trying to see, we're looking at deeper spike-specific repertoires to see if there are more such antibodies where clonotype-restricted mutations are indeed driving maturation and are indicative of binding to atypical or cryptic epitopes like M15. Um, so finally, I'd like to thank the members of Coelho, Byte, Simon, Kramer, and Wiesman Labs, who were major collaborators for our work on M15 that got published in Cell Reports earlier last year, and Dr. Ali Alibedi's lab for our current ongoing collaboration and my thesis advisory committee. And again, Immcantation for giving me this opportunity to present. Thank you so much, and I'm happy to take any questions.